and Dr. Julie Segre from NIAID is giving that. And Julie's background is that she has a lot of work in genomics and in skin biology. She trained, got her PhD at MIT in Dr. Lander's lab, and then trained at the University of Chicago on skin biology. Joined NIH in the Genome Research Institute, where she is now as a senior investigator. And Julie's going to talk to us about the skin microbiome. Thank you, Julie.
Thank you. Okay, so normally the last speaker says, I'm the only one between you and the bar, but I'm the only one between you and this fabulous meeting being over. So I decided that I would just filibuster instead. I'm just going to talk, and then you won't have to leave. And this is my hometown.
I'm setting my watch now.
Okay. So it is true. I've been part of the Human Genome Project for 20 years, and lots of things to me look like a nail that I could hit with my hammer. Because I really do think that DNA sequencing is a powerful microscope for microbial identification, and I also think that microbial genomics is a field that translates incredibly well. The information that we can generate with genomics, and I'll try to focus on that, are lessons that clinicians can use fairly rapidly. And I think it's really important, as we even talked about earlier today, that we incorporate clinicians and scientists into these projects so that we have translational research built into the project and can immediately find the avenues where this information could be useful. So a number of years ago, my lab, along with my fabulous collaborator, Heidi Kong, so any of the scientific ideas you can say are jointly between me and Heidi, and all the editorial comments are just my own appreciation of the field. So we looked at the skin microbial diversity and found that there was tremendous diversity of the bacteria on the skin. So that really my left arm is most similar to my right arm in the bend of the elbow, but the bend of my elbow is more similar to the bend of your elbow than it is to my underarm, because this is a hairy niche with big hair follicles that's very sweaty, and this is a moist surface. So we've studied for a long time, and we continue to study the skin microbial diversity. And here you can see the similarity of the moist sites that are in green, the oily or sebaceous sites in blue, and the dry sites in red, and that there are these predominant bacterial phylum, but there is greater diversity than appreciated, and that this is sort of a topographical map. And what I was saying here, you can see that if I just looked at 4 individuals, that their back would be predominated by Propionibacterium, but the antecubital crease, the crease in the elbow, would be predominated by these proteobacteria. There can be outliers who are in a state of microbial dysbiosis because this person could be staph colonized. That orange is the Staphylococcus. Recently now, we've completed our fungal diversity. And so we had said that we were doing this microbial survey, but really we were just doing bacteria. And I really want to second David's comments from earlier today. We need to start thinking about bacteria and fungi. But to do this, we needed new technologies. As David said, the first thing that happens, if you don't lyse it, you don't sequence it. So we spent time working with different fungal cultures and trying to find a protocol that would lyse all of these fungi. And then we tested whether the 18S rRNA or the internal intervening transcribed sequence, which is between the 5.8S and the 18S, what would give us the best resolution. We then created an ITS database because there is a 16S database that we all use for bacteria, but we didn't have the resources to do this with fungi. And that's been preventing people from looking at fungal diversity in the way that we look at bacterial diversity. So we built this ITS database, which is perfectly coordinated with clinical microbiology labs that use the ITS sequencing also, and we developed phylogenetic classification to the genus and species level. To begin to understand whether this sets the stage for considering skin disorders. You know, that eczema is always on the inside of the elbow and psoriasis is on the outside. Does this topographical resolution that I have of the skin begin to predict why there might be microbial involvement for these disorders, and that it might be very specific to the disorder? Because dermatologists have known for a long time that part of the diagnosis of a rash is about where is it on your body. And I also wanted to get to a few other issues that we've sort of played around with here. I wanted to talk about age and disease state and how they alter the skin microbiome. So first of all, with age, we've spent a lot of time talking about newborns. And we've talked about how newborns and how you can't reset after that. Well, one of the things that fell out of our data analysis that I was totally shocked by was that when you go through puberty, there is a dramatic shift in the microbial community. So I'm showing it to you here quantitatively, and then I'll show it to you qualitatively, which will actually probably impress you more. But when we look at the principal components analysis, Tanner is a staging of where you are. Tanner 3 is sort of on the edge of puberty, so this is sort of 11 to 13-year-olds, maybe 12 to 14-year-olds. They have a very diverse microbial community, but it is completely distinct from kids who have gone through puberty, which does actually conform to a norm more so than even the diversity that we see amongst prepubescent kids. And so here's what you can see of the difference between puberty, and you can really call this from across the room. What you see here is that the early Tanner stages are rich in these proteobacteria, this red up here, and there's really not that much actinobacterium in green. When you've gone through the transition through puberty, it's all about the actinobacterium. I've just shown here, this is actually the Nares, which you wouldn't have expected inside the nose. But this is true of the Nares and skin sites, that there is a sharp demarcation and transition as you go through puberty. I just say this in that we've spent a lot of time talking about, you know, the first 5 minutes of life and the first year of life. But there are other transitions that we do go through as humans, and there are perhaps other points that we need to think about that you could reset your microbiome, and that there are other places that we also need to think about. What is the, you know, how are we providing medical advice to people, and what, you know, why are we going through these transitions, and, you know, the extent to which, you know, we either promote this state of health. But you can see these are really dramatic shifts where the amount of Corynebacterium goes from 10% to 65%, and the Propionibacterium, which are the lipid-loving bacterium, go from, you know, 0 to 20. So not surprising that you get a greater percentage of bacterium that live on sebum as you go through puberty. We've talked about atopic. We've talked about the hygiene hypothesis. We've talked about atopic dermatitis a little bit. You've seen pictures of eczema. But I want to give you my own take on this, because I think one of the things we haven't talked about is the use of the skin microbiome as a biomarker. So Heidi and I have a clinical trial that enrolled kids with moderate to severe eczema. And really the interesting thing here is that, you know, the hygiene hypothesis, well, as the human geneticist, I would say if these disorders have increased in the last 30 years, and for atopic dermatitis, it is clearly not the diagnosis that has changed. These diseases are increasing. And 50% of the kids who have, you know, severe eczema will go on to develop asthma. There's a genetic link to a skin barrier protein that is not expressed in the lungs. It is really about this epicutaneous sensitization. But if there's a change, our genetic states can't change that fast. I can't change and evolve my 23 chromosomes and suddenly start having, a different genetic state. But I do agree with Marty. Our microbial ecology could have changed in those 30 years. And like my husband would like to see what Chicago looked like 500 years ago, I think it's very important to look at what is, you know, our microbial state before we started taking all of these antibiotics. this is what atopic dermatitis looks like. It affects the inside of the arms and behind the knees, and the kids will progress. Like they'll be, at baseline, but then Heidi scores each patient. This is the AD severity. Then they'll be given treatment, and it'll resolve. And this is a busy slide, but I'll just tell you that what we're really seeing here is that during the flare, there's this increase in Staphylococcus, and in particular, the pink down here means that this is Staph aureus. After they've been treated, and we see that the microbial diversity is even somewhat returning, even if they have the symptoms, they look like they're in a flare. If they have used some treatment, they're already in a greater microbial diversification. The bacteria that are present post-flare are all things that we can find in the baseline, The controls, even if the kids do have Staphylococcus, it is rarely the pink Staph aureus. It is more commonly the blue Staph epidermidis or the other types of Staphylococcus. So what we are seeing here, this is a disease that's episodic, kids, but the parents are searching for the cause. Why is my kid fine? And then one day, the itching starts, they don't sleep, I have to put them, what is the trigger here? And what we can say with genomics is, what we see is that the diversity, when you have atopic dermatitis in a flare, the diversity just plummets, and the amount of Staph aureus just dramatically increases. So you can see also that when the score of atopic dermatitis is greater, the amount of Staph aureus is greater. So from this, with genomic studies, what we can do is develop these hypotheses. But this is really where we need to be, is I'm going to say that this, when we think about how we're going to use microbial data, we're going to use it in the context of biomarkers. I am not going to come up with a cure for atopic dermatitis directly with this research, but there are a lot of intermediate steps that we should be looking at, in that many parents would like to know, when is my kid going to go into a flare? And if I wait and see that the kid is, scratch themselves red, it takes a while for that flare to resolve, because by now you've got this enormous, excoriated skin. What about if we could start to intervene here in the pre-flare state? Is there some possibility that these kids travel along at baseline, and then we don't know what happens first? Does the microbial diversity drop? Does the proportion of Staphylococcus increase? But really, could we begin to use this drop in microbial diversity or the increase in the levels of Staphylococcus as a biomarker? Like the kids take their, blood glucose levels, could we begin to say, your microbial diversity is dropping. Now is the time that you should be stepping up the treatment. You need to be putting on your creams every night when you get out of the bath. And if you could get it that you could predict when the kid was about to have a flare before they got to the part where they had scratched themselves, yes, actually that would be providing some benefit. So we don't have to wait until we're all the way through these stories to come up with the mechanism if there are biomarkers that we could use based on the microbial diversity to tailor therapies for patients or to predict who was about to have a skin flare. Okay, we also looked at the fungal diversity of these patients, and it's the same story as the bacterial diversity. It's now a more permissive, open niche. And you do see fungi here. I mean, with the primary immune deficient patients, we are also looking at the fungal diversity because that's part of the medical management of them. So within atopic dermatitis, I think we've looked at, we can't answer what is the driver, but I think if we could even just back it up and get some biomarkers to know when a kid is about to flare, that still would be useful. And hopefully long-term we would start to understand what is the driver of this disease, what makes some kids more likely to be colonized with Staph aureus. But I don't think we should only look to the future without thinking about what are the goals that we could accomplish with this data right now. Age affects the microbial composition. Again, I don't know what it is about going through puberty that changes your microbial composition, but I think that's another time point that we need to think about, not just looking at the first year of life. and the ways in which the immune status does alter the microbiome. So we've looked at these patients with very rare primary immune deficiencies, but there are a lot of people in the U.S. and in the world who are made immune compromised either because of an underlying hematologic disorder or because they're being prepped for transplant. So the idea of how the microbiome changes during an immune compromised state is something that we do need to consider, because those infections that occur during the immune compromised state are often a lot of how the disease needs to be medically managed. In terms of future directions, I think metagenomics, we consider really 16S and ITS. These are sort of stepping stones. We, technology is bringing us where we can have data that just looks at all the genes encoded by in these sites. The problem is there that we get these data sets and 30% of the genes are, open reading frame, function unknown. Well, we need to start solving the function of more bacterial genes so that we can begin. Otherwise it looks like, at the level of metabolic state, all these bacterium are very similar. But that's not true. It's just that the part of the genomes that are really interesting have not been annotated. As this conference has explored that there are other microbes that we haven't considered yet. I think the metagenomics will hopefully get us to the viruses. And finally, this is my lab, but as I told you, Heidi Kong is an independent investigator, a dermatologist who works with me. Much of the work that I showed you was done by Elizabeth Grice, Julia O., Keisha Finley, and I'm sorry I didn't have time. Well, I did. This is a separate story about our hospital-acquired infections that Evan Snicken has worked on. And Joy developed the fungal ITS database, and I really do hope that people use that. We'll certainly be, we'd like to make that, available, and we'll help everyone to get that installed. And we really hope that people can start to do fungal bacterial joint analyses. So thank you.
Well, thank you, Julie, and all the speakers this afternoon for a great session. We do have time for one or two questions if there are things very specific about Julie's presentation, and then I'll ask everybody to come up and maybe have a little discussion. Any specific questions? Yes, Marty.
Julie, tremendous tour de force. I noticed one thing in a few of your slides that it was very interesting to me. We kind of saw it on a smaller scale before, and that had to do with malassezia.
Yeah.
And most clinicians, the only malassezia they've ever heard of is malassezia furfur, which I didn't see in your slide, nor did we have in our studies.
Right.
That's the one that's easy to grow. The other guys can't grow.
Right. Thank you, and if that's how you pronounce it, I'll try to remember that, malassezia. We were able to identify that there are, of the 14 known malassezia, we found 11 of them. That's a compressed view of the data. We did see the malassezia furfur, but it's not one of the predominant malassezia species, so we just, in that figure I just showed, 4 of them. You're right. I think it really gets us to this interesting idea that there are common commensals, but then they're also associated as pathogens. And if you want to grow malassezia, you just have to overlay your plates with olive oil.
Julie, I have one question. Usually we are seeing in Peru chronic infection dermatitis in children, and we don't know, usually we culture Staph aureus, but we don't know if the Staph aureus produces some toxin to produce eczema, or the eczema is contamination with the Staph aureus. What is your opinion? Because that is very similar to your model you have right now.
Right, and actually I think that's a level of resolution that we hope that we'll get to with the metagenomics, because when I say Staph aureus, I don't know whether it is the entire Staph aureus population that is increasing, or whether at the baseline they have multiple different Staph aureus genomes, and it is just 1 or 2 that actually increases during the flare, and then that's what is causing the flare. So we're hoping that when we do metagenomic sequencing, we'll be able to resolve how many different Staph aureus species were there at the baseline, and are we selecting for just a few of them during the flare? And Marty told me how to solve that problem, but it actually was complicated. We were supposed to look at the ribotype of just the Staph aureus and look at their 16S full length gene, but it got complicated because Staph aureus, like Staph epidermidis, has 5 copies of the 16S RNA gene. So we couldn't resolve whether it was just a few of them or if it was multiple. But I think that's exactly the next step that we need to know, is whether there are certain types of Staph aureus that are causing this. Because again, that's part of what we're seeing with many diseases, is that there are variants that are commensals and variants that are pathogens. And we've seen that with Staph epidermidis when we sequenced a lot of genomes. There are specific genomes that we consider professional commensals, and we never see them, as causing infections on indwelling medical devices. But that's I think that's the next level of resolution.
Just a quick question. Any changes in gender relative to these or during puberty with females versus males?
So we didn't see any differences in the skin microbiome that was relative to gender. We do have the caveat that it's that maybe we need larger studies. But I think, gender is one thing, but maybe it's also determined by your place of birth, or where you lived for the first year of life. I mean, there are so many possible confounders, but we didn't see a signature of gender, and we certainly looked for one because every time we submit a paper, people ask us that.
David?
Julie, thanks for all your wonderful observations. Just a quick note. Your point about the importance of strains and strain variability, highlighted in a paper I know you know, Journal of Investigative Dermatology from a group at UCLA that did this with P. acnes isolates from acne and non-acne skin and saw trends towards, sets of genes found in the acne-associated isolates that were distinct from the gene set in the non-acne-associated strain set.
One quick question. There's no evidence, though, that antibiotic therapy is of value in atopic dermatitis, is there?
Well, there's tremendous, that is the mainstay of treating atopic dermatitis, is topical. So we have a number of different therapies that can be used for atopic dermatitis. We just don't know which one to use for which patient. It looks like one disease, but it's probably 4 or 5, diseases that all just are classified as one disorder because they all look the same. But it is true that pediatricians will, say, we'll try this, and if it doesn't work, come back, in 3 weeks. And that's part of the frustration, is that I think we would have better uptake if a physician had some way of distinguishing, this all looks like eczema to me, but you're of a microbiome type that you're going to respond well to topical steroids. And they don't try you with 2 or 3 things that are not going to work. And they say, and it'll resolve it should resolve, so don't even bother. And, but what we're really looking at is, but does that then predispose the kids to developing more severe asthma and allergic rhinitis? Maybe if the physicians thought that they were affecting a long-term outcome, that they would be more aggressive in the treatment.
Mm-hmm. But does eradication of staph reduce the risk of all these episodes? Or I'm just a little unclear about that.
Okay,
Yeah.
so, so you mean whether prophylactically?
Getting at what is this an etiologic role for Staph aureus or not?
Oh, look, hopefully Dr. Kong is going to the microphone to answer that question.
Okay, and one other related
No, she's
question.
going to tell you the answer.
Yeah, I was also going to ask, what about the staph superantigens, and do they have any role in this?
Oh, I'm sure Heidi's going to answer that, too.
Hi. So it's interesting. There are a lot of these patients, and there is a heterogeneity to these patients. Even though for our studies I was very clear in phenotyping, because there is a wide range of eczema, but these patients were fairly cleanly phenotyped. And even within that group, you can see that there is some variation. I do want to note that our population that you see, those changes were not necessarily a result of antibiotics. Only a small proportion of those patients actually received systemic antibiotics. Most of those changes were affected through maybe more aggressive topical steroids or use of topical bleach baths, which is used, has been shown to have some effects in this disease. So there is a strong microbial association with the disease. And even in patients, we've observed, and you can see it in the primary immunodeficiency patients, that they are managed with chronic antibiotic use. And yet, even if they are on chronic antibiotics or go through pulses of antibiotics, they can still get flares of their disease. So there is something, definitely, Staph aureus bacteria are playing a role in this, but there is something else going on as well. So it's a complex disease. And the superantigens, people have looked at the superantigens, and it's not really conclusive exactly what exactly is going on.
Thank you.